Hello, biologists. Today's lecture is about population ecology. So what is population ecology? Population ecology is trying to understand how a group of one species exists in its life, right? So we're going to look at populations, population density, dispersion, and changes through time. Each one of those things has subcategories under it, so we'll get into it. The example I wanted to start with was the known areas of wolf activity, and this is in 2015. There are some updated ones, but I like the diagram for this one best because it shows us the wolf management boundary, which is this red line. This is our state of Oregon. We've got a scale bar, and we've got our denoter of which way is north. Then we have known areas of wolf use, and each OR number is the name of the wolf and its pack. So this is going to be the OR28 pack. This is an unnamed pack. This is the Untamilla River. Here's the Walla Walla pack. And so then if we go back to our key, if it's blue and hashed, it's the estimated area of wolf use. If it's gray, it's status unknown. So we've got a gray one down here, the Keno pack. And then if it's red, wolf packs are no longer present. So this is where they used to be, but are no longer there. So we use this data to try and predict what the wolves are doing, how healthy the population is, whether they're moving or expanding, just to try and keep in watch on what's going on. And that's the goal of population ecology. Here's a little bit more detail about how we can get into what population ecologists study. For size, you're looking for the total number of individuals present. So it's the individual number of wolves, or in this example, I have a map of several continents. We have a lot of red area, and we have some areas of blue. And this is the historical in red and blue present distribution of lions. So if we are looking at lions, we want to know the total number of lions, but then we want to figure out the density. The density is the total number of lions per unit area. Like all of this historical area might have been where they were found, but what if 30 of them hung out right here? So the density per area can be totally different. Think about humans in cities, higher density than humans in the rural area. So we live everywhere across our country, whatever country it is, but we are pocketed in places where we have a higher density. Dispersion is how our organisms are arranged spatially. So are they clumped together like we do in cities? Are they uniformly dispersed? And I have a diagram of this coming up. Or is it kind of random? Do they just grow wherever their seeds fall like a plant? Then we also have occupancy. In that last diagram, I showed you areas where we're not sure wolves are found, where we know they are no longer found, and where they are currently found. So that's also related to population distribution. That's what this diagram is showing you, historical use and present use. We want to track both because it tells us something about that population. And we also look at geographic range. So this map is showing you geographic range too. What can we infer from this? We can infer that lions were found in a lot wider area historically than they were currently. What are the reasons why? Well, we could speculate, but until we start collecting data and figuring out what's going on, we won't fully know. Here's another diagram showing us some population information. So this is going to be Australia. And in Australia, we are looking at the density of kangaroos. So imagine that you wanted to study kangaroos. If this was our range, we would just look at the hash line here, and we would know that we can find kangaroos anywhere within this area, excluding these two islands. But if we wanted to increase our chances of actually finding a kangaroo to study, we would want to know the density because we want to go to areas of greater density to try and have a better chance of finding them. Yes, kangaroos are found here, but the density is so low, it might be one for several kilometers. And so here, we're going to have a lot greater chance of identifying them and finding them. So how do we do this? How do we figure out who lives where and how do we try and study them? we have three main ways that we do this: quadrats, transects, and mark and recapture. First, let's talk about quadrats. You're going to be doing the lab using quadrats. A quadrat is just a square. That's all it is. They can be a meter, they can be a foot. People even use round ones, but then that kind of defeats the word quadrat. But it is a piece of material that has a certain shape size inside of it, and that helps you determine what's going on in your population. So on the right here, I have a PVC quadrat. It's made of PVC piping. It's square. It is probably 1 meter by 1 meter. That's the most common way that we make quadrats. And this one has wire going across it to help you see the spaces inside in smaller amounts. How do we use this? You put it on the ground and you count and identify the individuals and the species inside your quadrat. This works because it causes us to focus on one area, and we can use this sample as a way to extrapolate what the higher population is in a larger area. Here's another visual of a quadrat. We've got two different quadrats. This one's PVC. This one looks like it's just pieces of wood. Again, you can make them out of anything. When you do your lab, you're going to see that I made some out of pants and I made some out of brooms. We just want a consistent size. So now you've got a consistent square. You're going to put that in several locations, identify the species and the number of individuals inside your quadrat, and we're going to use that to extrapolate something. Either extrapolate the population density or the population range or what kind of trends are going in your population. But the ultimate goal is that it gives you a place to focus and collect your data, because it's not usually possible to collect the data from something like an entire habitat. So if I was curious about the types of plants that were in a park near my house, it would be impractical to try and count and identify every individual species and organism in that species in the entire park. Instead, my quadrat is a representation of it. So here, this is an intertidal pool quadrat sampling, and they are collecting little snails. Next, we have a transect. A transect is basically just a line, and you collect data along that line. So here I have a visual of it. I've got a woodland, and I've got a lake or a pond or the ocean, a body of water. And I'm curious how my plants change between these two boundaries. Are the plants the same the entire way across? Are there species that live here that don't live over here? Are their numbers the same across? Is their density the same all the way across? And so a transect, it gives me a line to focus on, because it's impractical to look at the entire habitat. We focus just on this area here. So when you're doing tree surveys, you do transect lines often, and you'll put a line through the forest that you're trying to survey, and you will say anything within 50 feet gets counted. And so you count all the plants on 50 feet on the right and all the plants within 50 feet on the left. And that gives you a representative sample of what the forest is like. Here's another intertide pool example. We have our transect lines, and these two biologists are looking at the species that are within a few inches of these lines, and those are the representative samples of this intertidal area. Lastly, we have what we would use for animals that can run away from us and won't sit still in our quadrats and transects, and that is mark and recapture. So in mark and recapture, we are going to take a sample of the population and we are going to mark them. This little mouse has an ear tag with its number. This bird here has a band on its leg. In theories, these don't cause any harm to the animal. They're just a way for us to track them, because visually we can't tell the difference between individual birds and individual mice. We can tell it's a bird and a mouse, and we can tell what species it is, but we can't tell 5 different mice apart in the field. We could probably identify one that has like a bald patch or a short tail, but the rest of them are pretty similar looking. And so we need to track them in some way. In mark and recapture, you're going to have the number of animals that you captured on the first day. Sorry, here. That's the M. And our speculated population size is what we want to figure out. So after you mark your animals and release them, you have to come back a second time and you're going to recapture your number of animals. And the total number you recapture is going to be divided, or you're going to divide the number that have markings by the total number of recaptured animals. And then you can use cross multiplication and rearranging this formula to figure out what N is, what your sample size is. It's not a perfect system, but it gives us a really good statistical estimate of how big a population is. Earlier, I said we want to look at the dispersion of our critters. This is what dispersion means. Some critters like to be uniform. For example, these penguins, they lay their eggs in their nests a distance away from each other so that they don't accidentally stomp on each other's eggs. And there are thoroughfares through, and so they end up pretty uniformly dispersed. Random is going to be usually wind dispersed seeds. So where these dandelions land is totally dependent on the wind, and wherever they land, they will grow. So there isn't a pattern here. And then we have clumped organisms that like to live in groups or communities or hives or cities. We usually see a clump dispersion for that. When we're tracking populations, we also care about their demography, which is going to be their demographics. And we're going to talk more about that in our next lecture. But particularly, we want to know their age structure. Is your population generally young? Is your population generally old? What is your age structure? You also have your sex ratio of male to female and female to male. That's important because if you do not have an equal ratio, or if you have very few females, your population might start to decline. So the number of opportunities for reproducing is denoted in our sex ratio. We also have our birth rate. How many babies can you have? And can you have more than one set of babies? How many babies you can have at once is fecundity. So the more offspring at once, your higher reproductive output, that's fecundity. And it's specifically at not once, not over your lifetime. So humans, we're not very fecund. We can do like one, sometimes two, rarely more than two, even though it does happen. We can have like up to octuplets. But for something like a cat, their litters usually contain 4 kittens. And so an octopus has hundreds and hundreds of eggs that it tries to lay at once. That is fecundity. We want to consider the death rate. Are we losing individuals quicker than we're really replacing them? And we also want to think about generation time. How quickly can you reproduce a new organism? Here's our graph of generation time. So we have body size denoted by length, and we have generation time, how long it takes you to replace yourself on average. Not like when do you become sexually mature, but on average, how often does each generation replace itself? So if we have a tiny little E. coli, it's got a reproductive time of about 20 minutes. Euglena, paramecium, these are bigger, but they're still microbes. We looked at these last term. These are our cute little protozoa. They have a reproductive generation time of about less than 1 day. Stentor and Daphnia, these are also microbes that we looked at. They have a reproductive time of less than 1 week. What's the point? We have a trend. So the bigger your critter gets, the longer the reproductive generation time is. So our tallest organism, our sequoia tree, is also the longest generation time. So sequoias will have a new generation roughly every 90 years. Birch, roughly every 80 years. Humans, we're a little over 10. This scale is not continuous. So we reproduce on average every 20 years-ish. It changes depending where you are in your cultural situation. All right. Now we mentioned whether or not you could have babies more than once. So birth rates greatly affect the population demographics. And some organisms can only reproduce once and then they die. For example, salmon and octopus, we call this simmelparity. And then if you can reproduce more than once in your lifetime, it's iteroparity. For example, these mammals here are lion and our cute little ground squirrel thing. We also want to track survivorship. So if you are born, your rate of dying is not equal regardless of what species you are. And so we can track your chances of dying with something called a survivorship curve. In a type 1 curve, you have low death rates early, but higher later. In a type 2 curve, it's pretty steady throughout. In a type 3 curve, you have high death rates earlier, but lower later. I think this is easier to look at in graph form. So here are our 3 types of survivorship curves. Here are our number of survivors. So higher up means more survivorship, lower means more dying. And then we have age, young to old. Note that it's not quantified because how long I live compared to how long an ant lives is very variable. And so we just have young to old to make it more qualitative. A type 1 survivorship has a high survivorship up until older age, and then everyone dies off. This is what humans have. We tend to have a really good survivorship up until, like, on average in America, people are living to like 76, which is really great. It's increasing too. So maybe in the next decade, our average will be up to 80. And then once you get old, your chances of dying really increases and your survivorship decreases. This is the opposite for ants. Ants have a hard time making it to youth, but once they make it to youth, they live a long time. Their chances of dying are higher when they are younger. So that is the type 3 survivorship curve. For a bird, there tends to be a steady chance of surviving its entire life. And so it is a type 2 survivorship curve, which is pretty much a straight line. You have an equal chance of dying at any time in your lifespan. I hope that was interesting. Email me anytime for questions, and we will chat soon. Bye.